So, welcome again to this study of mineral nutrition. Next, we have gone through the different macronutrients which are required by the plants in higher quantities, and their concentration is also comparatively high. Contrary to this, micronutrients whose concentration is little bit low, is relatively small in the plant and required by the plant in minute quantities for growth and development and overall health of the plant. The first micronutrient in this category is iron. So, iron is the second most abundant metal in the earth crust after aluminum. So, it is an iron also serves as essential micronutrient with numerous cellular functions, and of course, its deficiency is going to represent one of the most serious problem in plants. So, just a quick overview of today's lecture, what we are going to study regarding the iron and other micronutrients. So, how iron is taken up by the plants? Plants generally obtain iron from the soil, typically in the form of iron sulfate or iron chelates. And iron can exist in two forms, uh, that is ferrous and ferric. You see, these are the two forms of iron which have limited solubility in soil, especially well aerated conditions where their concentration remains below 10 to power minus 15 molar, and which make them less accessible to plants at physiological pH levels. But soils with alkaline pH and bicarbonates often lack generally sufficient iron. So, what is the way out? How to make availability of iron for uptake? So, it need to be modified in the soil and through a process facilitated by detachment from the mineral soil particles. And plants basically employ two main strategies to mobilize and absorb iron. So, you see in the slide there are two strategies, strategy 1 and strategy 2. So, first we will discuss in detail what is strategy 1. So, you see that in the slide that in strategy 1, which is very much prevalent in most monocotyledonous and dicotyledonous plant except grasses. So, iron acquisition is basically regulated by availability of soil iron, by the availability of iron in the soil and the development of stage of plants requiring iron. Under iron deficient conditions, plant generally enhance the activities of specific transporters and enzymes. So, this strategy involves the acidification of soil, reducing the pH from 8 to 4, which increases the concentration of ferric ion from 10 to the power minus 20 to 10 to the power minus 9. So, then root plasma membrane bound proton ATPase, that is shown in the figure, facilitate this reduction in the pH. So, basically ferric ion is then reduced to ferrous ion, that is Fe2, by a plasma membrane bound ferric reductase oxidase before being transported into the root cells by iron regulated transporter 1, that is IRT1, which is an iron regulated transporter. So, now we will discuss, you see in the second slide, there is second strategy, and this strategy has been adopted by the members of the family Poaceae, namely barley, rice and maize. And basically this strategy relies on the chelation of iron, ferric ion basically, with low molecular weight phytosidophores, particularly of the muginic acid family. These siderophores bind these ferric ions in rhizosphere, making it accessible to the roots. This chelatin based mechanism allows grasses to efficiently utilize iron from iron deficient soils. The ferric and PS complex is then transported into the uh, epidermal cells of iron deficient roots by high affinity transporters. So basically in strategy second, high affinity transport plays a very significant role, and you see that overall the overall maintaining the iron homeostasis is very crucial for the plant, as iron is necessary for synthesizing heme, iron sulfur prosthetic groups, and then assembling into apoproteins. So iron is stored in the plastids in the form of phytoferritin, a protein molecule capable of enclosing approximately 4,500 ferric ions. So this strategy second is also important, but limited for certain plants. Now, what are the specific roles of iron in plants? So generally iron is required in large amount as compared to other micronutrients, but you see that it is essential element for the synthesis of chlorophyll and carotenoids. Iron is also very important component of cytochrome, ferrodoxin, flavoprotein, formation of chlorophyll, porphyrin, activation of catalase and peroxidase enzymes. Uh, and in addition to this, iron is also a very vital component of the iron sulfur clusters present in the very important enzyme nitrogenase. And basically these clusters serve as a cofactor that facilitate the transfer of electrons during the conversion of atmospheric nitrogen to ammonia. And in addition to this, specifically iron participates in the electron transfer reactions essential for the reduction of the nitrogen gas. In addition, that nodule formation is a complex process involving signal exchanges between the plant and rhizobia, and iron containing proteins and enzymes are involved in various signaling pathways that initiate the formation of nodule in response to the presence of compatible rhizobia in the soil. So the what are the prominent heme proteins that are present in plants? The prominent heme proteins are the cytochromes, which contain an iron porphyrin complex as their prosthetic groups, you see in the figure. And you see that cytochromes are integral to the redox system within the chloroplast, mitochondria, and participate in redox chain of nitrate reductase. Other heme enzymes include catalase, peroxidases, which generally exhibit reduced activity under conditions of low iron availability in plant tissues. In addition to this, you see that generally the enzyme catalase helps in detoxification of hydrogen peroxide to water and oxygen, and playing a crucial role in the process such as photorespiration, we have gone through into details, and the glycolate pathway, as well as in association with the superoxide dismutase. Now, in non-heme iron sulfur proteins, iron basically coordinates with cysteine thiol groups or inorganic sulfur as clusters, or it could adopt both ways. And ferrodoxin is a well known iron sulfur protein acting as an electron carrier in various metabolic processes accordingly that have been shown in the figure. Uh, like isoenzymes of superoxide dismutase contain an iron sulfur, which is a metal component involved in detoxifying superoxide anion free radical. So, another enzyme aconitase, which contains iron sulfur protein, catalyzing the isomerization of citrate to isocitrate in the tricarboxylic acid cycle. In addition to this, iron is also very necessary for the stability and activity of these enzymes, though they may not also contain copper, zinc, manganese or iron as a part of metal component. So, in this way iron is very much important for the plant, and if there is any sort of deficiency, that will be reflected in the plants. So, what are the specific deficiency symptoms uh, in plants? And basically in plants, the deficiency iron could develop due to the insolubility in soil rather than its absence. So, insolubility plays very important role. Generally, optimum concentration of soluble iron for most plant ranges from 10 to the power minus 4 to 10 to the power minus 8 mole, with slightly acidic soils contains optimal amount. Soluble iron concentration of 10 to the power minus 9 m or lower generally is very common in calcareous or alkaline soils with low bioavailable iron are insufficient for plant growth and could lead to leaf chlorosis. So, you just try to recall whether iron is mobile or immobile in plants. So, generally iron is immobile element in plant, hence deficiency symptoms include intervenal chlorosis and necrosis initially appear in the younger leaves because of being immobile. Unlike magnesium deficiency, where symptoms usually start in older leaves, but the iron deficiency symptoms are first visualized in younger leaves. So, intervenal chlorosis, which appear generally in the between the veins uh, and result if deficiency continues for longer duration, the entire leaves turn yellow, and in most severe cases young leaves may turn white with necrotic lesions. You see in the figure, this is the plant of cannabis which is deficient in iron. So, you see that in this plant deficiency of iron could be visualized initially in the form of intervenal chlorosis, but at later stage the whole cannabis leaf become chlorotic. Uh, Generally, major symptom in the once plant is deficient in iron, it could lead to the inhibition of development of chloroplast, and generally the deficiency of iron is very common in the members of family Rosaceae like maize, sorghum and other fruit trees. Now, what are the toxicity symptoms of iron? So, generally iron toxicity occur rarely, but generally in case of iron toxicity the leaves become bronze colored, you see in this slide, and brown spots uh, generally indicate the deficiency of sufficiency, more than sufficiency, or you can say toxicity of iron, and generally this condition is very much prevalent in rice leaves where toxicity could be easily observed, and this brown colored leaves are often referred as bronzing, and this could lead to a significant challenge in crop production, especially in waterlogged soils, where it could be ranked as the second most severe factor limiting yield in wetland rice cultivation. So generally in wetland rice cultivation, this toxicity is very common, and the damage caused by iron toxicity is primarily attributed to the formation of reactive oxygen species. And generally, once plant faces these oxidative stress, plants activate basically crucial cellular defense mechanism. And one such mechanism you might recall involves the induction of antioxidative defense enzymes like ascorbate peroxidase, which generally help to mitigate the harmful effect of reacting oxygen species. And in addition to this, plants could employ iron binding proteins such as ferritin to sequester excess iron and could minimize the toxic impact on various cellular processes in plants. So these defense strategies you see that serves as a vital role in safeguarding the plant against the damaging effect of iron toxicity, ensuring the optimal growth and productivity, particularly in case of rice. Now, next essential mineral element is chlorine, and you see that chlorine is also a very important micronutrient required by the higher plants, and particularly it participate in several physiological processes and involved in various growth and developmental processes, particularly maintaining the osmotic and osmotic stomatal regulations, as well as in evolution of oxygen in photosynthesis. Generally, this collide is readily taken up by the plants, and its mobility in short and long distance transport is very high. And basically, chlorine occurs more prominently as chloride ion in the soils and plants, and it is very much abundant in the form of inorganic anion present in the plant cell. Moreover, generally higher plants could contain concentration of chlorinated organic uh, compounds, and their number could be up to 130. Now, as like other essential mineral elements, generally plant absorb chlorine primarily in the form of chloride ion from the soil through their roots. So, chloride basically enters plant roots through specialized transporters, transport proteins, and these proteins are basically embedded in the roots and cell membranes. And these chloride transporter in the plant cell include CLC protein family, CLC, that is chloride channels, which include both chloride channels and chloride nitrate proton antiporters and cation chloride transporters, which likely function as sodium potassium chloride co-transporters. So you see in the figure that these transporter basically facilitate the movement of chloride ions from the soil into the root cells, and once iron reaches inside the roots, these chloride ions are transported within the plant through the xylem, reaching different parts of the plant such as leaves, stem, and reproductive structures. Now, what are the specific roles imparted by the chlorine in plants? First, you see that at cellular level, iron this uh, chloride you see that functions as is at the for regulating various uh, metabolism, particularly in the formation of cytochromes, uh, which is involved in various cellular processes. You just recall in the light reaction of the photosynthesis, this chloride acts for a very important role in the formation of photolysis of water. So chlorine, you see that generally very essential for plant nutrient and generally required by the plants in very small quantities for optimal growth and development. You see in this figure that chloride ions are involved basically in the process of oxygen evolution within the photosystem. Second, likely it exert influence on the structural and function of oxygen evolving complex, and basically this complex is located within the photosystem 2 and facilitate the oxidation of water to generate oxygen. Within the PS2, colloid ions are basically situated near the entrance of potential proton transfer pathway, suggesting that it plays a role uh, near the uh, within the PS2, you see that colloid ions are situated near the entrance of the potential proton transfer pathway, suggesting their mm, involvement in the release of protons from the uh, magnesium oxide calcium cluster. The water oxidation cycle within the PS2 typically requires one or you see two colloid ions, and studies have generally shown that depletion of colloid ion inhibits transition between S2 and S3 as well as between S3 and S0 states you see in the slide. And basically this colloid ions play a role in the water oxidation complex of PS2, particularly binding to manganese or calcium atoms, regulation of redox potential of manganese oxide uh, calcium cluster, or could be involved in maintenance of hydrogen bond network and in the activation of substrate water. So in addition to this, if you see these in the slide that chlorine also contribute to the, we have previously discussed a very important aspect that chlorine also contribute to the regulation of stomatal opening and closing, which influence the gas exchange and water uptake. For example, in plant species like Allium cepa, it is onion, which do not generally synthesize malate in their guard cells. So at this juncture, chloride is very essential for stomatal functioning and stomatal opening, which is generally inhibited in the absence of colloid ion. Additionally, chlorine is also involved in osmotic regulation and ion balance within the plant cells. So basically various cations and anions generally stimulate membrane bound proton pumping ATPases and uh, phosphatases, various pyrophosphatases. The proton pumping ATPases, they are located in the plasma membrane, is generally activated by monovalent cations, with potassium ion being particularly very effective. However, the proton pumping V type ATPases found on the endomembranes, especially stimulated by chloride ions and other ions. So here you see that how chloride ions is playing very specific role in proton pumping. Now, what are the specific deficiency symptoms induced by chloride in plants? So you see that plants deficient in chlorine, whose leaf you see on the slide, that leaves, they are they are necrotic and chlorotic and necrotic, and generally severe deficiency could lead to curling of youngest leaves followed by shriveling and necrosis may occur. In addition to this, chloride deficiency also could lead wilting of the leaves. Wilting of leaves, especially at, at the leaf margin, uh, is indicative of chloride deficiency. And in addition to this, premature senescence of leaves found in fracture and stem cracking are typical symptoms of deficiency of chloride. Chloride deficiency could also lead to swollen root tips. In general, the deficiency of chloride, you see that could lead to stunted root growth, and generally it affects the cell division, particularly cell extensions, which are generally impaired in deficient plants. And you see that these in the slide, and the leaves of tomato plants is showing a specific deficiency symptom reflected in the bronzing of leaves, which is chlorosis and necrosis as present on the leaves, that could be well visualized. Now, what are the specific deficiency symptoms of specific toxicity symptoms of chloride? So, chlorine basically generally pose chlorine toxicity could pose a worldwide challenge and serves as a notable stressor that could hamper the plant growth, particularly in soils in arid and semi-arid regions of the world. Generally, when the concentration of chlorine increases to 20 millimole in the external solution, then plant species become susceptible and they are very much prone to chlorine toxicity. And conversely, you see that there are certain species which are tolerant to chloride toxicity, and these species they are, you see that they are very much resilient, uh, enduring high concentration of chloride that could be 4 to 5 times higher without compromising the growth. So these discrepancies of in chloride toxicity threshold predominantly could result from the variation in the sensitivity of leaf tissues to elevated chloride concentration, and also could be due to the genotypic differences. Uh, so thank you very much for today's session. On the next turn, I will discuss function of other essential mineral nutrients. Thank you very much.